Welcome. In this session, I'm going to discuss about sensory organs. The various sensory organs in human body include nose, tongue, eye, ear, and then skin. And of course, here skin is the largest organ in the body. So here, nose is meant to sense smell. So this is olfaction. Then tongue is meant for sensing taste. So this is gustatory. And eye is meant for vision. Then ear is meant for hearing, so that is auditory. Whereas skin, it receives various stimuli like touch, pressure, temperature, etc. So these are various sense organs in the human body. Now we will see the first one, nose. So let's say about nose. Inside the nose, it contains mucus coated receptors, and these mucus coated receptors are specialized for receiving sense of smell. Okay, and these mucus coated receptors are called olfactory receptors. So these olfactory receptors are bipolar nerve cells. So olfactory receptors are bipolar nerve cells, and these bipolar nerve cells will be about 100 million. So those cells are present 100 million in the olfactory epithelium. So let's see the diagram of the olfactory epithelium. The olfactory epithelium consists of three types of cells. So it consists of three types of cells, and those are olfactory receptor cells, and these are olfactory receptor cells. Just now I said they are bipolar nerve cells. And then the next one are supporting cells, and the third one will be basal cells. So these are three types of cells which are present in the olfactory epithelium. So in this picture we can see from this point to, okay, so this point it extends the olfactory epithelium, and here these green colored ones are the olfactory receptor cells. So these olfactory receptor cells, okay, will have olfactory cilia. So these are called olfactory cilia, and these olfactory cilia are embedded in the mucus coat. So here you can see an yellow color. So this is the mucus coat, and this mucus is produced by these Bowman's glands. So there are Bowman's glands located in the epithelium, and these Bowman's glands will produce mucus. Now these olfactory receptor cells are supported by a number of cells present around them, and these are called, okay, so you can see here the supporting cells. So these supporting cells, okay, support these bipolar nerve cells. Now at the base of this we have again the basal cells, right? So here all these are olfactory receptor cells, which are actually bipolar nerve cells, right? And I told that there are around 100 million bipolar nerve cells which are present in the olfactory epithelium, and there, okay, one end will have olfactory cilia which will be embedded in the mucus, and you can see here these are molecules which are received by, okay, the nerve endings, that is olfactory cilia, and from here, from each of the olfactory receptor cells is the olfactory, okay, nerve fibers that will join the olfactory bulb. So these bipolar nerve cells directly extend into olfactory bulb, so that is present behind it, right? So in this picture you can see the location of olfactory epithelium. So here is the location of this olfactory epithelium, and you can see the nerves straight away enter into the olfactory bulb. So these olfactory bulbs are extensions from forebrain. So they are extensions from forebrain, right? Okay, so this is the location, and how these olfactory receptor cells are located, and from them the nerves directly enter into the olfactory bulb. So here is the opening of the nose. This is nostril, okay, so through which the odor molecules enter inside, and they come in contact with this right olfactory epithelium. And the olfactory bulb is connected to the limbic system. Bulb is connected to limbic system. So that's about the nose. Now we'll see about taste buds on tongue. So the next sensory organ we'll see is about tongue, and we know located on the tongue are taste buds. So here let's see the structure of these taste buds. So this is the surface of the tongue, and here there will be a small pore. So it's called a taste pore, through which actually the food substances will come in contact with the right, the gustatory cell. So there is an extension of these receptor cells. So here these gustatory cells are the receptor the receptors. So there are the gustatory receptor cells. So the taste buds contain gustatory receptor cells. So these gustatory receptor cells will receive. So here the one which I have mentioned here in red color are the gustatory receptor cells, and they are supported by uh, certain cells, right? So you can see here, and from them arises are nerves. So here there are three nerves involved, okay, in receiving the information. So all those are cranial nerves. So cranial nerves are involved in receiving the information. So they are the facial nerve. We know the facial nerve is seventh cranial nerve, and then the glossopharyngeal nerve, and this glossopharyngeal nerve is the ninth cranial nerve, and then vagus nerve. So this vagus nerve is the 10th nerve. So these nerves will carry the information to the brain, right? So that's about taste buds on the tongue. So next we'll see about eye. So eye is involved in vision, and uh, we have one pair of eyes. So one pair of eyes, and these one pair of eyes are located in orbits of skull. Here the human eye, it starts developing from third week during the embryonic development and continues up to 10th week. So it starts in the third week and continues up to 10th week, and the cells from both ectoderm plus endoderm will contribute in formation of eye. So they contribute in the formation of eye. The adult human eye is almost spherical in shape, is spherical, right shape, and has a diameter of around 2.3 centimeters. Has a diameter of 2.3 centimeters, okay, and the wall of eye is composed of three layers. So those three layers are called sclera. So this will be the outer layer, and then there is a choroid layer. So this will be the middle layer, and the innermost layer is retina. So this is the inner layer. So here when we see eye, right, of course the eyes are covered by upper and lower eyelids, and here in the corner of the eye we find a small bulb like structure, and this is called plica semilunaris. So this plica semilunaris is a vestigial of nictitating membrane. So this nictitating membrane is functional in case of lower animals. The lower or animals like birds and reptiles, right? In them you will find this uh, nictitating membrane. Now in case of human beings it is completely non-functional, and we call it plica semilunaris. Okay, so here is the cornea of the eye, and so this is the iris we see here, and the rest is white here, and this is called sclera, and this portion is cornea. Okay, and these the rod like structures what we see them are iris, and at the center is pupil through which light enters into the eye. So now let's see the diagram of eye. So here I'm showing the LS of eye, that is longitudinal section of eye, and this side it is anterior side, and the other side is posterior. Is the posterior side. Okay, and we can see here there is a layer sclera, the outermost layer, that is sclera, and this sclera on the front it becomes the cornea. So you can see here from this point to, okay, this point is the cornea here, so the remaining will be the sclera. So let's see about the sclera. So this sclera is a dense connective tissue. So it is made up of dense. It's a connective tissue which is dense connective tissue, and it's commonly called white. Commonly called white, and is composed of right, the collagen and elastic fibers. So composed of collagen and elastic fibers. So I told you that the anterior portion of the sclera is called cornea. So you can see here all this from this point, okay, to this point, it's called cornea. So there is a junction between cornea and the sclera, and that what we call it the corneal limbus. So you can see here a small, okay, point. So this is called corneal limbus. It's called corneal limbus, and this corneal limbus is the border present between cornea and sclera. So here in this picture I can show this is the corneal limbus. Is the corneal limbus. Okay, and this cornea is covered by a membrane, a mucous membrane. So you can see here there is a membrane that covers this cornea, and this is called conjunctiva. So this layer that covers the cornea is called conjunctiva. So this conjunctiva is a mucous membrane that helps in, so conjunctiva, a mucous membrane that helps in lubrication of eye, or lubricating eye. So right, so that's about the sclera, right, and the cornea, and that is covered by conjunctiva. The next one we shall see about the choroid layer, which is a middle layer, and this choroid layer, it consists of many blood vessels. So it contains many blood vessels, and it will be bluish in color. It is bluish in color. Here the choroid layer is thin on, so you can see here the choroid layer is thin on the posterior 2/3 of eyeball. Okay, so it is thin on okay, posterior, right, okay, 2/3, so 2/3 of eyeball. Now here the choroid layer will become thicker on the anterior side. So you can see the choroid layer extends here. So the choroid layer extends here, and then that it becomes the ciliary body. So it becomes ciliary body. So you can see they have mentioned here this one. So from here to here, all this is the ciliary body. So the ciliary body is extension of the choroid layer. So choroid layer on anterior side, so it forms a ciliary body. It forms ciliary body. Okay, and the ciliary body itself will continue forward to form a pigmented area. So you can see here the ciliary body, it extended forward here. So you can see here this is the portion that extended the ciliary body, and this becomes, okay, the iris. So here the ciliary body will continue forward, and it forms a pigmented and opaque structure, and that pigmented opaque structure is called iris. So this portion which I have mentioned here is iris. So here, not a confusion, this is not this portion is not, okay, aqueous chamber. So aqueous chamber, so this all, okay, the front portion that is present in front of the lens is the aqueous chamber. I'll come to that later. So this is aqueous chamber actually, and this is iris. So you can see here this is how it looks. This is iris. Okay, right. So the ciliary body extends much forward on in front of the lens, and it forms, right, a pigmented and opaque structure. So here pigmented and opaque structure. So that pigmented and opaque structure is iris. Now the ciliary bodies through ligaments. So these ciliary bodies through ligaments hold the transparent and crystalline lens. Hold transparent and crystalline lens. So you can see, okay, here lens, and here I'll show you. So this is a small portion, so that extends, okay, and it connects to the lens. The ciliary body will connect to this lens, and these are called the suspensory ligaments. Called suspensory ligaments. So you can see the suspensory ligament on both sides. So you can see the ciliary body extends forward. I told that it forms in front of the lens the iris, and also it holds the lens with the help of suspensory ligaments. So these suspensory ligaments are also called zonules. So suspensory ligament is also called zonule. Right. Next one. So here the iris is not completely covering the lens. So there is a portion you can see, right, that is left out. So here and to here. So this is pupil. So this is a pupil, okay, and it is through which the light enters and falls on the retina. Here, the diameter of the pupil is adjustable. So, depending on the intensity of light, the pupil diameter increases or decreases. So that's what, when there is no light, or say suppose we are seeing in dark. So in the dark, okay, the pupil dilates, so it becomes bigger or widens to allow maximum light to enter the eye, so that there is a visibility. So the opposite will be in bright light. So in bright light, the pupil becomes narrower. So I'll write here. So pupil widens or dilates in, okay, dark, whereas the pupil, okay, decreases its diameter, decreases in diameter in bright light. So here in the night time or in the dark, okay, the pupil widens or it increases in diameter. So in this case it is increases in diameter to allow more amount of light to enter the eye, so that there is a greater visibility. I mean, to increase the visibility. Next, coming to retina. So this is the innermost layer. Okay, so you can see here, this is the outer layer, that is the outer, the sclera, and below that is the second layer, that is choroid layer, and this red colored one is the innermost layer, that is retina. So now let's see about this retina. So here, the retina again has three more layers. So it has, okay, three layers, and those three layers are: one, there is a ganglion cell layer, ganglion cell layer, and another one, bipolar cell layer, and the third one is the photoreceptor cell layer. So these are, okay, three layers: the ganglion layer, then bipolar cell layer, and photoreceptor cells. So this will be from, okay, inner of this eye to outside. So the ganglionic layer will be present here, the ganglionic layer, so we will be seeing here. Okay, so the next to that will be the bipolar cell layer, and the photoreceptor cell is almost present to the choroid layer, whereas the ganglionic cell layer will be facing the vitreous chamber. So here, in the eye we have two chambers separated by the lens here. Okay, so here, in front of the lens, there is a small chamber, it's called aqueous chamber, and behind the lens is the vitreous chamber. So the vitreous chamber and aqueous chamber are filled with fluids, and they help in maintaining the diameter of this eye and keep it touched all the time. If the liquid from the vitreous chamber is lost, then the shape of the eye is lost, and the lens cannot focus exactly on the retina. So hence, the fluids are very important, right? Whereas in case of this vitreous chamber, the fluids are almost present, and they are not replaced, whereas in case of aqueous chamber, the fluids will get replaced, right? So we'll see here, the ganglionic cell layer. So this contains ganglion cells, contains ganglion cells, and then bipolar cell layer it consists of the bipolar cells. So these are neurons. The ganglions and bipolar both are neurons. And coming to these photoreceptor cells, the photoreceptor cell layer, it consists of cells called rods and cones. So it consists of cells called rods and cones. Okay, we'll see the rods. The rods actually help in the night vision, whereas cones help in day vision. So that is scotopic, and this is, that is the day vision, it is called photopic. Right. So of course the twilight and the night vision so will be assisted by these rods, whereas the cones will help in day vision. So of course even the cones are responsible for visualizing colors. So the rods are not involved in detecting the colors. So only the cones are the one who help in detecting the color. So here there are three types of cones. The cones for blue wavelengths, so that detect blue wavelengths. So cones that detect blue wavelength, I'll simply say cones for blue, and then cones for red, so they will detect the red wavelengths, and there is cones for green. So the cones for blue, they detect the blue wavelengths, and the cones for red, they will detect red color wavelengths, and green for green colored wavelengths. Right, I will discuss this retina layer again in detail. So coming back to the eyeball, so we can see here on the retina at one point, so there is a right yellowish pigmented spot, right? So this yellowish pigmented spot is called macula densa. I'm sorry, macula lutea. It's not macula densa. So here I'll just show the region. So here is the macula lutea. So this macula lutea is a yellowish pigmented spot. So this is yellowish pigmented spot, and right at the center of that yellowish pigmented spot, that's called macula lutea, or macula lutea. So this is a yellowish pigmented area. So at the center of this yellowish pigmented area, that is macula lutea, right, there is fovea. So that is within this region is a small depression. So you can see here there is a small depression, and that small depression is called fovea. And here the fovea is a region, actually it is fovea centralis. So this fovea centralis region is where the number of cones are present in large numbers. They are present in large numbers. Okay, so that's why we say fovea is a point where the visual acuity, that is the resolution, So the resolution, we'll call it visual acuity, so is very high or is very much, that is greatest. So the resolution is greatest at this fovea centralis. So this is the fovea centralis. Okay, so you can see, so this is a small scooped out region in the macula lutea region, which is a yellowish pigmented region. Okay, so of course the yellowish color of the macula lutea is due to presence of certain substances, that's the pigments like right xanthophyll carotenoids. So there may be presence of this xanthophyll, okay, and carotenoids, I mean xanthophyll carotenoids, and there will be lutein, okay, and then zeaxanthin are different pigments that are present in this macula lutea, which gives its yellowish coloration. And little away from this macula lutea region, we have is a blind spot. So this blind spot is a region from where the optic nerve arises. So you can see here, this is the point, the blind spot area. So this is the blind spot area, right? So this is the point from where the optic nerve arises and carries that information to the brain. Okay, and it is also a point where the retinal blood vessels enter the eye. So the point where the optic nerve, okay, along with that there is an entry of retinal blood vessels. So the blind spot, right, is also a place where retinal blood vessels enter the eye. And one more point about this macula lutea. So this is oval shaped. So this is oval shaped and has a diameter of 5.5 mm in diameter. So the macula lutea is oval shaped and it is 5.5 mm in diameter. Coming to this aqueous chamber. So this aqueous chamber is filled with aqueous humor, is filled with the aqueous humor, whereas the vitreous the vitreous chamber, so this one is filled with the vitreous humor, right? So this vitreous humor is a viscous fluid. It's a viscous fluid, and we can also call this vitreous humor as vitreous body. And the major substances which are present in this viscous humor will be, I mean the viscous fluid will be albumin. So vitreous humor has albumin, then it also has hyaluronic acid, so etc. are the substances which are present in this vitreous humor. And as I told that vitreous humor, it actually helps in right maintaining the shape of eyeball. So this vitreous humor help in eyeball, right? And in case of this aqueous humor, so aqueous humor is formed by, so this is formed by ciliary processes, is formed by ciliary processes. And the formation of this aqueous humor, it is continuous. So the formation is continuous, whereas the vitreous humor, it is not continuous, is only once it is formed, okay, inside the vitreous chamber, and there is no time to time replacement of vitreous humor, but there will be change in case of aqueous humor, so it will be replaced, right, uh, from time to time. Next, coming to the part of lens. So this lens is a circular biconvex transparent body. Is a biconvex transparent body, so which is enclosed within a capsule. So here, so we'll see like this is the lens here. Okay, so on either side I told that it is suspended by the suspensory ligaments. So these are suspensory ligaments. Okay, so here this is the anterior side of the lens, and it has, okay, lesser curvature on anterior side, lesser curvature on anterior side. Coming to the posterior side, it has, okay, greater curvature on the posterior side of the lens, and this one actually helps to refract the light and helps in focusing the light on the fovea. Okay, and here the adjustment of this lens, the adjustment of a lens to focus an object, to focus an object is what we call it accommodation, is called accommodation, right. And here one more important point here, lens has no blood supply. So there is no blood supply to lens, okay, but it gets the nutrients from the aqueous humor. It gets nutrients from the aqueous humor, and moreover it has a low metabolic requirements, so, and also low metabolic requirements or low metabolism. So low metabolism is seen in case of lens, and the nutrients, even though it has no blood supply, the nutrients it gets from the aqueous chamber. And lens also doesn't have nerve connection. It also doesn't have nerve connection, right. So no nerve connections we'll see in case of lens. Okay, so in the with this I'll end up today's session. So in the next session we will see more about the retina and those layers, the ganglionic, bipolar, okay, and the photoreceptor. Even we'll see the structure of photoreceptor cells in the next session.